In this lecture on functional connectivity, we'll be looking at the source of the resting state fMRI signal. What is the evidence that resting state functional connectivity is neuronal in origin? Can resting state functional connectivity reflect individual differences? Resting state functional connectivity was discovered in 1995, and there were a handful of publications each year, and then it really took off in the mid to late 2000s. And one question is really, why did it take so long for this resting state connectivity to really explode and take off in the field? Well, a lot of the reasons for that is that in the first several years, there was a lot of skepticism about whether these resting state fluctuations were real. That is, whether they really reflected synchronized neuronal fluctuations. And this is reasonable to ask because, after all, we are saying that areas are functionally connected if the signal in two parts of the brain go up and down together. And there's a lot of other reasons, in addition to task-related or spontaneous neural activity, that the signal could be going up and down in a synchronous manner. There could be physiological noise, just the heartbeat and respiration. It could be subject motion, scanner instabilities, and so on. So in those first years of resting state fMRI, there were a lot of really great studies laying the foundation and showing that these fluctuations truly are neuronal. First, there was a study from Dietmar Cordes, actually from right here at University of Wisconsin, where he looked at what frequencies contribute to the correlation between these different regions. And if you look at the auditory, visual, and motor regions, you note that it's mostly the very low frequencies that contribute to the correlation, below 0.1 hertz. But if you look at arteries, veins, or cerebrospinal fluid, it's mostly dominated by higher frequencies, 0.1 to 0.5 hertz, which overlaps with respiration-related frequencies, or 0.6 to 1.1 hertz, which relates to the cardiac frequencies. So it's unlikely that these are just driven by the heartbeat and respiration, that we are really looking at much lower frequencies. Next, Scott Peltier found that if you look at the echo time dependence, typical task activation shows a linear increase in the percent signal change with increasing echo times, and resting state functional connectivity shows exactly the same pattern. In addition, Brad Biswell in 1997 showed that the resting fluctuations decrease during hypercapnia, that is, if you give somebody a higher proportion of CO2 to breathe. This is actually the same thing you see during task activation as well with the bold response. And the reason for that is that CO2 is a very potent vasodilator. And so if you give somebody CO2 to breathe, it increases their resting blood flow and actually drives out some of that deoxygenated blood. And so there's less change that you see during the activation. Another piece of evidence why resting connectivity is neuronal is in this study from Quigley and colleagues, also from here in Madison. It turns out there are some individuals who are born without a corpus callosum, called callosal agenesis. And in a typical healthy control subject, you would see nice bilateral connectivity. That is, things on both the left and right show strong connectivity. However, in these patients with the callosal agenesis, you see a somewhat asymmetric pattern of connectivity. Another piece of evidence why resting connectivity is neuronal comes from a study from Johnson and colleagues, where they had the opportunity to study somebody with resting state fMRI before they were and after they underwent a callosotomy. That is, where they cut the corpus callosum in order to treat epilepsy. And you can see before the surgery, you can see nice bilateral connectivity between these different brain networks and regions. And after the surgery, you see that becoming much more asymmetric and not showing that bilateral pattern. In addition, there are a number of resting state networks that we consistently observe, and they match with other known functional networks, such as the motor network, the visual network, and so on. And so to argue that this is just a motion artifact, or perhaps this is just due to the specific characteristics of the blood vasculature, you would have to argue that the blood vasculature is different in visual cortex than for the default mode network, which again is different from the auditory network or from frontoparietal networks and so on. And that becomes a little bit hard to defend that for some reason the vasculature is so specific to each of these different functional networks. Another really great study showing that this is indeed neuronal in nature is from Krienan and Buckner. And here they looked at both motor cortex connectivity and prefrontal connectivity, but they asked for every voxel in the brain, is it more strongly connected to the left motor cortex or the right motor cortex? And what you can see here is that the left cerebellum is more strongly connected to the right motor cortex and vice versa. The left motor cortex is more strongly connected to the right cerebellum. And this is actually something you would expect based on how the brain is wired. That is, the left side of your cerebrum controls the right side of your body, but then the cerebellum, that's flipped. So it is actually the left side controlling the left side of the body. And this is not something you would expect based on the vasculature. The cerebellum and the main cerebrum have different blood supplies. And finally, there have been studies where they've acquired simultaneous functional MRI and EEG data, such as this study from Laufs and colleagues, where they looked for are there areas or signals in the functional MRI data that are correlated with the fluctuations in the EEG. And indeed, they did find regions where the fluctuations in the fMRI signal at rest were correlated with the fluctuations in the power EEG signal. So I hope I've demonstrated that the resting state functional MRI signals that drive this measure of connectivity truly are neuronal in origin. However, how useful is it? That is, can we see all of the brain networks during the resting state? Can we map individual differences in the connectivity? This is a really great study from Steve Smith and colleagues. And what they did here was they performed an ICA decomposition of both the brain map database and resting state fMRI. So the brain map database is actually a database where when people have done task activation studies, they report coordinates of where they've seen activation, and then they submit this to the brain map database. So this database contains thousands of studies of areas that are generally active in different kinds of tasks that were studied with fMRI. And so by doing an ICA decomposition of this database, they can find what areas of the brain generally co-activate together. And when you look at that in comparison to resting state fMRI, an ICA decomposition of the resting state fMRI, you can see striking similarities, suggesting that really with resting state fMRI, we can find the exact same networks that people have been finding in general in the literature that co-activate during all of these different task activation studies that have been examined. In addition, there's some evidence that these intrinsic fluctuations actually can account for some variability in the behavior across different trials. Also, I would recommend for a nice overview and review of particularly some of the early work in resting state fMRI, this paper from Mike Fox in Nature Reviews Neuroscience. What about individual differences? Well, there's been a number of great studies on this. Here's one of the early ones showing that looking at the default mode network in individuals with autism spectrum disorder compared to typically developing controls, that there are differences in the connectivity of the default mode network. In this study from Adriana DiMartino and colleagues, they showed that within a typical community sample, there were differences in functional connectivity that were related to trait-like characteristics, in this case, the social responsiveness. There's also been evidence that resting state fMRI could be used to guide treatment strategies. For example, finding the best site for transcranial magnetic stimulation in order to help the antidepressant efficacy. And then also some research that I've been involved in, where we found that the resting state functional connectivity of the amygdala and the hippocampus with this prefrontal cortex region was correlated with the amount of childhood trauma as reported from the childhood trauma questionnaire. In another study, we also showed that early measures of cortisol, which is a stress hormone, that were acquired when the individuals were 4.5 years old, were actually correlated with the resting state connectivity later when the individuals were 18 years old. And finally, there are a number of large Connectome projects that are currently going on. One of these that just finished collecting data is the Human Connectome project, the Young Adult Human Connectome project in particular, where they've acquired data from 1,200 young adults, and this is available to any researcher. In addition, there are ongoing studies now looking at development of functional connectivity, looking at the connectomes in different disease. We had 2 of these here in Madison, one looking at Alzheimer's disease and one looking at epilepsy. And finally, there's also a very large study called the Adolescent Brain and Cognitive Development Study that has just recently started. This is a study where they've recruited about 12,000 9 and 10-year-olds, and they're going to be following them over the next 10 years, getting imaging measures every other year. This study is being acquired at about 20 sites all across the country, and this data is actually available to researchers to use to investigate. So I think it'll be really interesting stuff looking and delving into this particular data set. So hopefully I've convinced you that the resting state fMRI signal truly is neuronal in origin and that it can reflect really interesting differences in behavior and brain organization.